Okay guys, this is Professor Bryan Yves Araneta. I am remaking my virtual molecular genetics lab. All this can also be accessed at the University of Utah Genetics Lab Center site. DNA extraction is a fairly easy technique given the right equipment and reagents. Now, why would you extract DNA? Here, it's needed in genetic testing, body identification, or analysis of forensic evidence. You can also add here paternity or parentage claims. And you may also want to extract DNA to identify a known species. For example, identifying microbes in a sample. Also, when doing genetic engineering or genetic experiments, you will also be constantly performing this technique. Now, we will be extracting DNA, of course, from cellular samples. So, tissues with nucleated cells, to be more exact, because you can only find DNA in the nucleus. Also, take note that this technique is usually done as a requirement for another lab procedure or a series of experiments and tests. Okay, so let's now get ourselves some cheek cells from our virtual subject. I know that in your zoology classes, you have experienced scraping the inner cheek to collect cells for your staining and microscopy activities. Let's follow these steps. And here are the things we need. Alright, now it's time to scrape some cells out. Let's place this in an Eppendorf tube. Let's add some lysis solution. This will break the cells in our sample to expose the DNA for extraction. And we will enhance this action by adding some heat. That's how things work at the molecular level. The detergent breaks the cell and nuclear membranes, and the enzyme proteinase K digests or breaks down the proteins called histones. We know that histones wraps our DNA strands. And we don't want a DNA still with protein wrappers in them, right? But of course, we still need to separate our DNA from the cellular debris. That means we must separate only the DNA from the other cell components, such as protein fragments, membranes, and other particles. You can check your chemistry text on how to separate mixtures. And we will be doing one common technique here. Let's add some concentrated salt solution first. The salt causes proteins and other cellular debris to clump together. This will make separation in the centrifuge easier. That's a counterbalance, of course, so it should be the same weight at the opposite end because we will be spinning this thing. Alright, so let's close the lid and let's go. Now it's done. All the other cell components settle at the bottom due to the spinning. DNA is fairly light and will stay in the liquid. Then we will now be removing this DNA solution by pipetting it out to another sterile Eppendorf tube. Let's add alcohol. And do some gentle shaking or agitation here. This will make the DNA visible. Let's go back to the centrifuge to separate the DNA from the liquid. And of course, after removing the liquid, you get your DNA extracted for the next steps in your experiment. Or you can store it for later use. I hope that's helpful, guys. Again, please subscribe to my channel. I'm trying to improve it now by replacing my old videos. I'm also trying to complete the series that I started, and I really forced myself to make daily uploads. Again, thank you very much for watching.